This video helps you revise the practical that shows the production of alcohol by yeast. In this practical, you need to establish anaerobic conditions. Yeast will respire anaerobically without the presence of oxygen to produce alcohol. Another name for this is called alcohol fermentation. It's really important that you can write the equation for anaerobic respiration. Yeast are anaerobically breaking down glucose to give 2 ethanol, 2 carbon dioxide and energy. In the lab, we set up 2 conical flasks, the experiment and the control. In the experiment flask, we put in yeast, cooled boiled glucose solution, a layer of oil, a fermentation lock, and in the fermentation lock we put lime water. The control contained the exact same materials except it did not contain the yeast. You will need to be able to explain how you made the experiment anaerobic. The first thing that we did was we boiled the glucose solution, and boiling glucose dries out the oxygen. Secondly, we added a layer of oil after we had added the yeast. The oil layer prevents oxygen from getting in. And finally, we added a fermentation lock on top of the apparatus. In the fermentation lock, we also added lime water. When CO2 reacts with lime water, it turns it milky. This occurs in the test flask only. This is because the control contains no yeast. Once the apparatus is set up, you need to place both conical flasks into a water bath for 24 hours at 30 degrees Celsius. After 24 hours, take out the apparatus and observe the results. You can see that something clearly happened in the test flask. You could see bubbles of CO2 in the fermentation lock and the lime water had turned milky. A question that has been asked in the exam is, how do you know that fermentation has ceased? And what you would say is there is no more bubbles of CO2 produced. The next part of the practical was testing for ethanol, proving that the yeast had anaerobically respired and produced ethanol. The test for ethanol is the iodoform test. The iodoform is made up of potassium iodide and sodium hypochlorite. The next thing you need to do is filter the solutions, so the test and the control solutions, and place the filtrate into a test tube. This should contain the yeast cells. Into the test tubes, add equal volumes of sodium hypochlorite and potassium iodide. Observe the results.